So what we're going to do today is last time with the idea of the macrophages finding their way to the site of bacterial proliferation. And those macrophages then would go into those sites and gobble up the damaged bacteria. They would gobble up the whole bacteria, and they would then process those bacterial proteins and convert them into fragments of those proteins. And we said that at the very end of the discussion that those proteins then, called major histocompatibility type 2 proteins, would then expose antigen fragments out on the surfaces of these cells. That's where we closed off. We're now going to see that there are two other related cell types that will be the so-called professional antigen-presenting cells. They come in the form of dendritic cells, which are really a kind of cousin to the macrophage, and another cell type we're going to spend some time on today, the naive or memory B lymphocytes. Those are the only three cell types that generate an MHC2 protein. They're the only three cell types that are so-called professional antigen-presenting cells because of their MHC2, and they will present fragments of antigens they've endocytosed. They will present those fragments to other cells, particularly T helper cells, and we'll talk our way through that. Before we can get too far down that line, though, we have to spend a little bit of time on what we mean by adaptive immunity in the B lymphocytes. The B lymphocytes, sometimes known as the plasma B cells, will secrete antibodies. These B lymphocytes are part of the so-called humoral branch of the immune system, and they're known as the humoral branch of the immune system because they're secreting those antibodies, those soluble proteins that are in the plasma of the blood and also secreted into the lymph. They're secreted into the so-called humors. Now, this is an old term from the Middle Ages, and the humors were blood and phlegm and bile, and they're just fluids. And so these B lymphocytes are secreting antibodies into the fluid, so it's into the humors. Then you'll remember we talked about T lymphocytes. T lymphocytes are those cells of the immune system that had to pass through the thymus before they became mature and effective. Well, the T lymphocytes come as two forms. They come as CD4 cells, also known as T helper cells, and they come as CD8 cells, also known as T killer cells. Now, the short version of this is that CD4 cells, also known as T helper cells, have transmembrane proteins called T cell receptors. The T cell receptors on CD4 cells recognize antigen fragments displayed on MHC2 proteins of professional antigen-presenting cells. So the sequence here is CD4 cell is a T helper cell. T helper cells can only talk to professional antigen-presenting cells because they can only talk to the MHC2 proteins. The other character here is that the CD8 cells are T killer cells. They too have a T cell receptor as a transmembrane protein. They too will recognize foreign proteins, antigens, on other cells. In both cases, both for the CD4 cells and the CD8 cells, these cells are restricted to seeing foreign proteins as antigen fragments displayed on other cells, either through something known as an MHC1 protein for the CD8 cells or an MHC2 protein for the CD4 cells. We'll walk our way through all of that, but the long and the short of it is the response of the CD4 cell, the T helper cell, is to release interleukins, which are like hormones, that will tell cells to divide and proliferate and make more copies of themselves. The CD8 cells, by recognizing foreign proteins on MHC1 proteins on other cells, and these will recognize foreign antigens on any nucleated cell. These CD8 cells now will, instead of releasing growth hormones, will now release molecules that kill that virus-infected cell, and we'll walk our way through how that happens. To understand the humoral branch of the immune system, we have to spend a little bit of time on what we mean by a secreted antibody and its structure. And this is a classic structure you've seen probably in a number of places. It has a classic Y shape. There's a long arm of the Y and a short arm of the Y. And we find that the antibodies are composed of a light chain and a heavy chain. There are 2 heavy chains and 2 light chains. The 2 heavy chains are identical to one another. The 2 light chains are identical to one another. But what makes these antibodies different from other antibodies is a region called the variable light region and the variable heavy region. These variable regions are at the short arm of the Y, and they have a particular 3-dimensional shape, which will be specific for a particular antigen. So the specificity of the antigen-antibody complex formation is dictated by the combination of the 3-dimensional shape of the variable heavy region and the variable light region. And we'll walk our way through that. And then we'll try to puzzle the idea of how these antibodies, in fact, protect us from foreign pathogens. And we'll see that these antibodies, once secreted into the outside world, will bind to foreign proteins. And in some cases, they'll cover over the sites that are toxic. In some cases, they'll cross-link the proteins, so the foreign proteins can be gobbled up by macrophages. Sometimes they'll cross-link bacterial cells that can then be gobbled up by macrophages, and we'll walk our way through that. Then the final 2 pieces here will be the interaction of T helper cells with professional antigen-presenting cells and the interaction of T killer cells with any other cell. Again, a T helper cell has to interact with a professional antigen-presenting cell. The professional antigen-presenting cell we'll talk about here are the naive B cells. We're going to find that these naive B cells are born in the red bone marrow, and they will have as a transmembrane protein a particular antibody with a particular 3-dimensional shape at the short arm of the Y. That particular antibody will be specific for only binding to one protein. Now, that naive B cell will then go out from the blood bone marrow into the germinal regions of the lymphatic tissues. Could be a lymph node, could be Peyer's patches, could be the spleen, and sit and wait. If that particular 3-dimensional shape of this protein on the naive B cell encounters a foreign antigen and binds to it, that starts a cascade of events. Cascade of events that looks a lot like what we saw for the macrophages, where the antibody will be, along with the antigen, will be endocytosed, passed through the lysosomal system, chopped into little pieces, put onto the MHC2 protein, and displayed in the outside world. Then after that, once it's displaying its antigen fragment, if it finds a T cell receptor on a T helper cell that makes a fit, then the T helper cell will then release growth factors, interleukins, that'll tell this naive B cell to divide and make like a gazillion copies of itself, and each of those cells will make the same antibody. Now, over time, after a few cell divisions, these antibodies go from transmembrane proteins to secreted proteins. So that's one of the changes. But then from this, we'll begin to see where memory comes from in the humoral branch of the immune system. So the naive B cell is only one cell, goes out into the world and proliferates. But after the cell proliferates, we'll store a few hundred or maybe a few thousand of these B cells, but we'll store them as memory B cells. So that then if we're exposed to that pathogenic antigen sometime later, maybe months later or years later, then that antigen will bind to the transmembrane protein antibody on the transmembrane protein antibody on the memory B cell, and that will start the proliferation all over again. And that's the reason you get a secondary response that's many, many times faster than the primary response. And then finally, we'll show the parallels between the T killer cells, which will use a T cell receptor and a CD8 molecule to recognize foreign proteins, usually viral proteins, displayed on another cell, and then kill those cells. We'll talk about how that happens. But let's begin here with a kind of reminder. And we'll talk about macrophages as professional, antigen presenting, cells. And this is where we were last time, where we had that macrophage which had found its way to the site of bacterial proliferation. It had endocytosed the bacterium. It had endocytosed the bacterium, put it into a vesicle, trafficked the vesicle to the lysosome, converted those proteins into fragments, usually no more than about 16 to 20 amino acids long. Then we have those fragments. The fragments then in the endosome, those fragments bound to this transmembrane protein here, called a major histocompatibility class 2 protein. We abbreviate that MHC2. Now remember, MHC2 is unique to professional antigen-presenting cells. This is one type of antigen-presenting cell, being the macrophage. We're going to see that there is a related cell called a dendritic cell that will do the same thing. And then finally, we'll see that there are naive and memory B cells that will also do this. And when we get this transmembrane protein out here as the MHC2, it now displays this antigen fragment. Macrophages do this. We're going to see a related description of this when we talk about naive B lymphocytes, with their antibodies being thrust out into the world, finding an antigen to bind to, and then running through this process. And we're going to see that MHC2 displays antigen fragment. Displays antigen fragments to T cell receptors on T helper cells, also known as CD4 cells. That's going to encompass a good bit of what we're going to talk about here. Let's take this panel, though, and describe antibodies of the humoral branch of the adaptive immune response. Antibodies are proteins. You can read glycoproteins secreted from mature plasma B lymphocytes. If you look at one of these mature plasma B lymphocytes, it's really chock full of endoplasmic reticulum. And the endoplasmic reticulum is heavily decorated with ribosomes. So there's a whole lot of rough ER. There's a highly elaborated Golgi apparatus. And there's a well-developed exocytosis, exocytotic machinery. And what these vesicles are doing is the vesicles are being loaded with antibodies. These are antibodies that once synthesized on the rough endoplasmic reticulum, processed with the Golgi apparatus, put into vesicles. When those vesicles fuse the plasma membrane, these antibodies are released, are secreted to the outside world. So hold on to the idea here that this is a characteristic of mature B lymphocytes, because we're going to come back to this in a moment and talk about naive B lymphocytes or immature B lymphocytes, and those guys have antibodies as transmembrane proteins. But hold on to this for the idea of what B lymphocytes do, what mature B lymphocytes do. They're secreting cells. And what are they secreting? Well, they're secreting this soluble protein called antibody. Now, the antibody has a particular shape, and you probably have seen some of this just in the general world, where they're shaped like a Y. Now, again, I'm going to draw this as a two-dimensional structure, but recognize as we go through this that because it's a protein, it has a proper three-dimensional shape. But when we look at this antibody, we find that there are two identical heavy chains. The two identical heavy chains are tied together by disulfide bonds. The disulfide bonds are nothing more than covalent linkages holding the two identical heavy chains together. Then out here in the short arm of the Y, there are these light chains. So in every one of these antibodies, there are two identical heavy chains and two identical light chains. Out at the end of the short arm of the Y, I'm going to put this in a kind of purple dotted line. Out here is the antigen binding site. Now, the antigen binding site is composed of a region of the short arm of the Y known as the variable heavy chain. We abbreviate this H sub V. Also out here is a region of the light chain called the variable light chain. The variable region of the light chain is called V sub L. Well, since they're variable regions, that implies that they're also constant regions. So this great long section up to the purple dashed line, this of the black is said to be the heavy constant region. And it's identical on both sides. But similarly, the light chain out here has a constant region called the constant light. This is the constant region of the light chain. So, to review, every antibody is composed of two chains that are identical to one another, the two heavy chains and the two light chains. All total, there are four chains, two light chains, two heavy chains. The two light chains are identical to one another, the two heavy chains are identical to one another. But now if we begin comparing antibodies released from different B cell lineages, we find what's different is out here at the short arm of the Y. And at the end of the short arm of the Y, there's a three-dimensional shape that's determined by the amino acid sequence, just as we've seen a million times before, where there's a three-dimensional shape at the short arm of the Y that determines the specificity of that antibody for the antigen to which it's going to bind. Let's have a look at a couple of pictures here and see if I can straighten that out. Now, this first image is this is an image which could have been drawn in about 1910, in the early days of immunology, when they began to recognize that over time, after an animal or a person was exposed to a foreign protein, was exposed to an antigen, there would be an increase in the concentration of antibodies in the plasma. So what this graph shows, on the ordinate, it's showing the concentration of antibody in the plasma. Remember, these are antibodies that are secreted into the blood plasma. And then on the axis is time. And in contrast to the time we normally think about in here in milliseconds and seconds, this is days. So one of the things I want you to see is at this point, this arrow says that's the initial exposure to an antigen. It could be a vaccine antigen, it could be that you get sick with a disease. And then you'll see that after the initial exposure, it takes some time before we see any change in the antibody concentration in the plasma. Then before we can get a peak of that antibody concentration, it's the better part of 2 weeks. So it takes a couple of weeks from the time you're exposed to that antigen until you produce a sizable amount of that antibody, that's antibody specific for the antigen, and have secreted that out into the extracellular space. Then you get over the disease, you throw off the infection, and the concentration of antibody comes back down to low levels. But now here at the second arrow, that's meant to represent the secondary exposure to the foreign antigen. It could be the primary exposure here was to a vaccine, and you've now generated an immune response. And the immune response now wanes after you get well again, but at some time later, sometime later, you're exposed to this foreign protein again, and you generate a secondary response in a matter of hours. This is the concept of memory. Remember, we said that the specific immune response has memory. This is where the memory comes from, and we're going to walk our way through the rest of the story to see how that happens on the molecular level. Yes?
So you'd mentioned exposure to a vaccine. Is that the idea behind booster vaccines then?
Okay, so I was going to try to make it too simple for the booster vaccines, but you're absolutely right. When you have a vaccine, normally what you do is you generate an individual protein. Usually it's one or maybe two proteins in a particular virus or some bacterium. And then you mount your immune response just to that protein, and it doesn't make you sick because it's just one or two of these inactivated proteins. When you get sick with a disease, then you have the whole panoply of those proteins from that virus or bacterium. And then you'll store away lots and lots of different shapes of antibodies to that whole panoply of proteins. But in a vaccine, because the number of proteins is limited, then you don't get as fully expressed an immune response. So typically there you have to have boosters. And I'm not sure whether they commonly use different proteins for the boosters or if it's just change in the concentration and you see it again and you boost the response. But that's why for most vaccines, you do require a booster. But if you get sick with the disease, you don't. And it's one of the reasons why probably we don't need to get boosters for smallpox, because smallpox is generated to a different kind it's a vaccine generated from a virus called varicella. And that varicella has a whole bunch of proteins on it that look like smallpox proteins. And so you probably are, those of you who have been vaccinated, you may be, because you may have been a first responder, but most of us haven't been.
Well, and the point several times, we got a live vaccination for
Well, the varicella, it's varicella? Doesn't sound right. What is it? That's chickenpox. Varicella is chickenpox. It's cowpox, and there's a name for it. And the cowpox then will then act as and that's a live virus, or it's at least an inactivated virus. And it's kind of dangerous, actually. People who are immune suppressed will actually get quite sick from it. And they don't like to give it to adults so much anymore. Used to be everybody got it before they were age 6. But since we've eliminated, presumably, the smallpox virus, we don't vaccinate anymore because the risk of the vaccine is greater than the risk of the disease. But at any rate, keep moving. This is the primary immune response, the secondary immune response, and this is meant to be the antibody response to the antigen. And again, we're going to see that these antibodies have this Y shape. And out at the short end of the Y, there is the business end of the molecule, which binds to the foreign proteins. One of the ideas that this cartoon's trying to get across is that any given protein will have numerous antigenic sites on it. Many, many sites on that one protein where we can raise antibodies. And in fact, typically the regions that will be recognized with the short arm of the Y are no more than 16 or 20 amino acids long. And so any given antigen will have a number of antibodies we can raise to it. And this cartoon is simply trying to say that there are three-dimensional shapes at the end of the short arm of the Y that will bind to those antigen, those bits of the protein. This is a two-dimensional image of the antibody that I drew on the board. So in purple are the heavy chains. These two heavy chains are identical to one another. In blue are the light chains. The two light chains are identical to one another. The heavy chains are tied one to the other by disulfide linkages. The light chains are tied to the heavy chains by disulfide linkages. Where we get variations in antibody structure is out here at the short arm of the Y. And that three-dimensional shape, differences in the three-dimensional shape, a result from differences in the three-dimensional shape of the variable light region and the variable heavy region. And that will provide a three-dimensional shape that will be specific for the antigen. Now, one of the things that cropped up, and when people started studying this, one of the interesting questions is how do we manage to make a nearly infinite variety of protein shapes so that we can defend ourselves throughout life against practically an infinite variety of potential antigens? How do we do that with a finite number of genes? And it was a puzzle for a long time, but I think one of the great discoveries of the 20th century was how that's done. And I'll briefly go through it here on this slide because I just think it's a miracle. And what happens is the naive B lymphocytes, and we'll talk about them in a minute, the naive B lymphocytes are born in the red bone marrow. And right after they're born, they start doing some mixing and matching of their genome. And what they'll do is they'll take one of these 300 variable regions, short segments of DNA, and they'll pull that out, and then they'll take another region of, yes, what do you want? Okay. What? Yes, what's your question about short chains, long chains?
Okay, so is the short chain always on the right, like on the board, but it doesn't look shorter in the picture? I just want to make sure that I'm not.
So this is a cartoon. If this were really a protein, then out here there would be a three-dimensional shape to this. This whole thing will have a three-dimensional shape. And we've simplified the three-dimensional shape just to show the linear structures of the heavy chain and the light chain. Okay? So that's all this is. So don't think that this is the true and absolute shape of an antibody. This shape is really not that much more accurate than that silly cartoon. And in reality, what we have is a three-dimensional shape out here, which is determining the antigen to which it will bind. And that three-dimensional shape out here is determined by this trick. And this is the trick where there are something like 300 variable regions in the genome and about 8 joiner regions. And the cell randomly chooses one of the 300, say 227. takes that 227, cuts out everything else, leaves only the 227 portion of the variable region, and then splices it in with one of the 8J regions. So now you get a whole bunch of variability simply from rearranging the DNA, and this then will produce an RNA, we'll splice the RNA, and we'll make a protein with this three-dimensional shape of the amino acids in the variable and from the variable and joiner regions out here, the short arm of the Y. That can be done, I think there are 3 different short chains you can do this with. You can also do this with the heavy chain, and we get even greater variability there, but it's the same story. We start combining things, and through that combination, we get an enormous number of degrees of freedom of the shape out here at the short arm of the Y. So that's where the shape distance differences come from. And then finally, what this is trying to say is how these antibodies now function. The secreted antibodies are going to go out and they're going to bind to antigens in the extracellular environment. And they're going to go out, in some cases, they're actually going to cover over the antigens of a toxic protein. So an example here where they're masking or covering over and neutralizing toxins. A great example of this is antivenom. If you go out walking on a spring day with your dog and your dog's off leash and your dog finds a rattlesnake, which is not so uncommon in these parts, and sniffs at the rattlesnake and the rattlesnake takes offense and bites it on the nose, then you've envenomated your dog. And these dogs can get pretty sick. So in some cases, you can take your dog to the veterinarian. They'll have on-hand antivenom. What antivenom is it's basically an antibody that's been raised in horses. And they'll take that antivenom and inject it into your dog and thereby cover over the toxic sites on the venom and prevent the action of the venom. More commonly, these three-dimensional shapes of the antibody will cross-link whole cells. So for example, they could cross-link whole bacterial cells. And these bacterial cells, once cross-linked, can be gobbled up by and destroyed by the macrophages. Similarly, we can do this with individual proteins. Proteins secreted by bacteria or individual viruses. We can cross-link them and then have them destroyed by the macrophages. This is a whole complement story, and we're not going to get into that. If you take an immunology course, you'll spend a lot of time on that. But that's basically how these antibodies function. They have a three-dimensional shape. They have a three-dimensional shape that goes out and finds the antigen and binds to the antigen. We'll have almost an infinite variety of these protein shapes because of that mix and matching of the genome. Because we can cut out one gene from the variable regions and the joiner regions and paste them back together and form a protein once we've gone through transcription, translation, with a particular three-dimensional shape out here. So that's the story of the antibodies. But the question we want to approach here again on this panel is the generation of a plasma B cell. And so we'll take this panel, and the story I'm going to tell you is pretty similar. It's going to be pretty similar to this macrophage story, but keep a watch out, and we'll talk about naive B cells, and clonal expansion of the protein. Let me say antibody they express. So now we're going to take this cell and we're going to call this a naive B lymphocyte. This is a cell that was born in the red bone marrow. It's born in the red bone marrow, and what it has done is it's generated a transmembrane protein antibody here. So this is a naive B cell that's just been born in the red bone marrow, and it's used that trick I showed you on the slide to generate a protein and put it out on its surface as a transmembrane protein. This naive B cell then leaves the red bone marrow and goes to lymphatic tissues, germinal regions. This naive B cell then, in the germinal regions, will have on its surface these antibodies. These antibodies are just a random shape. We just pretty much selected them randomly from that rematching of the genes we talked about a moment ago. Then these cells have a three-dimensional shape on this. They're out in the world, and they have a window of time. They have about, 10 days to 2 weeks to bind a foreign protein specifically. If they never find that foreign protein, if they don't bind anything out here in 10 days to 2 weeks, they die. But if they're in the lymphatic tissues, in the germinal regions, and the single lone cell binds to antigen to form an antigen antibody complex. The binding of the antigen to the antibody starts endocytosis, just as we saw basically for the macrophage. It starts the process of bringing that antigen antibody complex into the cell. The vesicle containing the transmembrane protein of the antibody with the antigen attached goes to the lysosome. The antigen and the antibody come apart, but now this antigen is converted to fragments. These are antigen fragments. These antigen fragments are probably no more than 16 to 20 amino acids long, so just short snippets of the protein that was bound to the antibody out here. Then, as we saw before, these proteins are passed into a structure known as the endosome. And in the endosome, the antigen fragment is attached to this protein called MHC2. Remember, there are only 3 cell types that express MHC2. They are macrophages, dendritic cells, and these B lymphocytes. But now, as we saw before, we now have an altar out here. And on that altar, we have displayed this antigen fragment. That's all well and good. But unless this B lymphocyte, which has endocytosed the proteins, passed the protein through its endosome and displayed it on an MHC2 protein, unless this cell is found by a T helper cell, nothing's going to happen. But if this cell is recognized by a T helper cell, then this cell is going to make like a gazillion copies of itself. And that's going to be the concept of clonal expansion. So I'm going to go to the upper panel again. And take this panel and label it as T helper cells and clonal expansion of naive B lymphocytes. So the character that we're going to start with is that naive B lymphocyte that we have on the upper panel. That naive B lymphocyte, which is serving as a professional antigen-presenting cell, is going to now interact with a T helper cell. So let's see how that's going to happen. So we'll start out over here with our naive B cell. This naive B cell, as we showed above, is a professional antigen-presenting cell. So that on its surface is this MHC2 protein displaying the antigen fragment that is the result of endocytosis of the antibody antigen complex. Now over here in blue is the next cell type in the story. This cell type is said to be a T helper cell. The T helper cell has another name. It's called a CD4 cell. And a CD4 is a transmembrane protein. And I think of the CD4 as a kind of lasso. I think of it as binding to the MHC2 protein. It doesn't bind to anything other than the MHC2 protein. It binds it, and what it's doing is it's bringing these two cells within a molecular distance of one another. It's basically mooring the two cells together so that a transmembrane protein that I'll show in green, the transmembrane protein here, composed of a beta subunit and an alpha subunit. Taken together, the beta and the alpha subunit are the T cell receptor. The T cell receptor has a three-dimensional shape out here at the end. This three-dimensional shape, if this three-dimensional shape fits the three-dimensional shape of the antigen fragment displayed on MHC2, what that will do is it will signal vesicles here in the T helper cell. These vesicles will come up to the membrane, and they'll, through exocytosis, they will release growth factors. These growth factors are called interleukins. There are a long list of interleukins. The one that's probably important here is interleukin 4 and maybe interleukin 5. The main idea, though, is that there's going to be on this cell, I'll put it in purple, on this naive B cell is an interleukin receptor. The binding of the interleukin to the interleukin receptor then signals to the nucleus. It tells the nucleus, start to divide. And this cell now will make thousands and thousands of copies of itself because of the signal from the T helper cell. This cell will make thousands of copies of itself, and each of the offspring of these cells will generate the same antibody. So we can do that as a kind of lineage over here. So that this first cell is meant to represent the naive B cell. The naive B cell divides into two. It's received the signal of the growth factor of the interleukin, and it divides. And that cell then has on its surface, as a transmembrane protein, these antibodies. And these antibodies are identical to the one up here. Then after a few cell divisions, we find that these cells now, after a few cell divisions, become plasma B cells. They secrete antibody. But the antibody that these cells are secreting from their vesicles has the same three-dimensional shape here at the short end of the Y as we had up here. We rearrange them a little bit, so we go from transmembrane proteins to secreted proteins. But their ability to bind antigen is still the same as we saw up here. And in this way, we take an individual cell, one single cell, and proliferate that cell through these cell divisions to make thousands of copies of themselves. And each one of those copies makes these antibodies. So that's this idea of clonal selection and cell proliferation. Well, that tells a pretty good story about how we can start out with one single cell for the primary immune response and generate the antibodies. But it doesn't tell us much about the memory. And remember that figure that I showed you said that, years later, we could get a secondary immune response. So the question that comes from that is, where does the secondary immune response come from? And that is, after we undergo this whole series of clonal expansion, we'll select some of these cells, a few cells, to be stored as memory B cells. These memory B cells then are stored, a few thousand of them, in the lymphoid tissue, so that if you're then exposed to the foreign protein at a later time, you can now use this same story and get clonal expansion and generation of the antibody. But where the naive B cell starts from a single cell, we can start now from several thousand of these cells. So that's basically how we get this idea of clonal expansion, and how we get the same antibody being produced by hundreds and hundreds of thousands of cells. Yes?
Is there notable difference between the memory B cells and the naive B cells?
Is there a notable difference between a memory B cell and a naive B cell? Well, one notable difference is that the naive B cell, if it doesn't run into antigen, dies in a couple of weeks. But the memory B cells will be with you the rest of your life. But they respond in pretty much the same way. I suspect there's slightly different interleukins. I suspect there's some subtle differences. But by and large, it's basically storing back tens of thousands of naive B cells that can interact now with T helper cells. And we won't even talk about it here, but their T helper cells undergo this clonal expansion too, so you get the proper three-dimensional shape of this alpha-beta subunit. And the expansion isn't quite as exquisite, but it's still pretty good. Yes?
Are they selected at random for memory, or are they
So a subset, and I'm not sure, and again, there are going to be another set of interleukins that will signal these secreting B cells to say, okay, revert back to a transmembrane state for your antibody and hang out and wait for bad things to happen again. And basically that's what we do. In the last couple minutes, let me finish this story. Let me do this on the backboard. And this is going to be the response of the T killer cell. Now, we'll start out here with a somatic cell. The somatic cell is any cell in the body that has a nucleus. And what these cells are doing is they're continuing throughout life. They're taking in proteins, cytoplasmic proteins. They're taking in cytoplasmic proteins, chopping them up into smaller pieces, and mounting them on a protein here called MHC1. Again, major histocompatibility class 1 proteins. All cells make these. And constantly what we're doing is we're taking fragments and putting them on these MHC1 proteins. Normally, these are cytoplasmic proteins, but if this happened to be a cell that had been invaded by an influenza virus, the influenza viruses we talked about earlier in the epithelial cells, then among the protein fragments that may be displayed out here would be a fragment of an influenza virus. And now I'll take you through the whole process here, but in the end, we have out here on the surface this transmembrane protein, the MHC1 protein, with a fragment, in this case, of viral protein. Now, normally this would be a self-protein, but in this case, we have a viral protein. And now along comes this cell, which is a T killer cell. It has a transmembrane protein again, called CD8. So T killer cells are CD8 cells. What the CD8 does is it moors this cell to the MHC1 protein, brings this cell in close enough. So again, we have these two proteins, the alpha and the beta subunit of the T cell receptor. Now, if there's a three-dimensional fit here between the T cell receptor and the antigen fragment, that again will mobilize vesicles. When these vesicles fuse the plasma membrane and release their contents, they release something called a perforin. The perforin then pokes holes for calcium in the virus-infected cell. And when it pokes holes in this virus-infected cell, it kills the cell, and this machine can no longer be a factory for the viruses. And this is a very similar story. If you hold on for just a minute, this is a similar story to what happened in World War II in Indonesia, where the Australians were trying to defend against the Japanese invasion, and they decided it was better to defend Australia and Indonesia and Sumatra than in Australia. So what they did is they hired a regiment of soldiers from the British Army called Gurkhas. And the Gurkhas were these outstanding night fighters, and they would go out into the jungles at night. They would jump into a foxhole and feel for the insignia badge that every soldier wears on their blouse, on their shirt. And if it was an Australian division, the Gurkha would say, "Good Aussie," and leave the foxhole. But if it was a Japanese insignia, he would kill the soldier. It's the same thing here with these T killer cells going out saying, "Good Aussie," if it's a self protein, and killing the cell if it's a foreign protein. And with that, I'll let you go.